The cancer cell therapy is a living therapy. Living therapy. Isn't that a great name for something which is really taking the cancer patient, here's your life, let's get on with it and return to our families and our friends. What began back in the 1950s and 60s as an allogeneic transplant is really becoming the cutting edge therapies that can really change lives. And the logistics are really one of the last limitations to allowing the patients to benefit from this life-saving therapy.
What is making it special, of course, the fact that you are working usually with patient-derived cells, from which you have to manufacture the product, or in some cases from some donors, but usually in all the cases it's about human cells that you are working with. Autologous therapies are based on the cells that you derive directly from the patient, so the product consists of the cells derived from the patient that you are then sending those cells back to the original patient.
The cell has to be alive, the cell has to be able to proliferate, the cell has to be able to be transferred into the patient and then to persist afterwards.
So the allogenic products are based on the donor cells from healthy donors. So you only have to culture the cells in a bigger amount, modify those cells, and then if there is a match in between the donor and the patient, you are able to support the patient with the treatment.
Right now, an average time from phoresis of the mononuclear cells to returning the product into the patient's body, that would be needle to needle, time should be in the range of about 3 weeks, okay? 3 weeks where the patient really has no therapy and the cancer is growing. And that 3-week time period is a stressful moment for the patients and their providers.
The challenges in this logistics part are simply based on the fact that we need to keep 30 hours from the start of leukapheresis collection, the collection of the cells from the patient, until the start of manufacturing. The manufacturing process itself takes about a week, depends on the indication.
In this case, we're actually individualizing every single shipment. You could imagine these materials have to travel in special containers. They have to be kept at minus 150 degrees, sometimes minus 20 degrees. We have to ship the material in a very short time frame to maintain stability. We have to make sure that the chain of identity between what we're extracting from the patient ends up back in the same patient. And how do we do that?
The patient expects to get back their own cells. Their life depends on that. If they're given somebody else's cells, they will be very sick. It could be life-threatening. In order to ensure the chain of custody, safeguards are implemented every time the product is moving from one person participating in that patient's care to the next. The checking and the double checking and the triple checking is critical.
So we at Marken are doing our best to design a supply chain in the clinical side to optimize the way we collect cells and genes from patients at their homes or in the clinic, get them back to the laboratory where they're being manufactured into a personalized medicine, whether they be allogeneic or autologous types of medicine, and then get them back to the patient in a way that protects the patient's privacy, protects the integrity of the sample, and the integrity of the drug product itself.
These are cells that need to be grown. They are cells that will die if neglected. They are cells that are stable if frozen. They are cells that can transform or change if not closely followed. This is not aspirin. And it's a partnership between the researchers, the clinicians, the patients, the pharma, and the government. It is time that we have these open discussions and that we educate each other, because the benefits are tremendous. This is the future of cancer therapy. There is no doubt.
And the cell and gene paradigm shift in our market is really the biggest step forward, I think, for us, for the industry, and for our families. All of these new investments that we're making are really preparing us for the future. I think about my kids. I really do believe in our lifetime and in the lifetime of our children, we will see major transformation